Okay, so thank you for inviting me. And I'm really happy to be here. It's been a fantastic conference thus far, and I'm looking forward to another day here. So what I've been hearing from most talks from yesterday, it's we know a lot about the biology of aging, and we know it in the different model systems, and we also know a lot in the clinical perspective, talking about frailty and sarcopenia. But there's not very much crosstalk between model systems and then humans. And also when it comes to neuroprotectors, so drugs that work to prolong lifespan or improving the health span, we know a lot what is happening in terms of mechanisms and drugs in these model systems, but we do not know a lot in the human. So there really needs to be some sort of translation in between here that's not really working at the moment. So I will try to come up with some ideas how we can bridge this a little bit. So then I would like to talk about biological age in the human setting. So what is biological age? Well, it's some sort of measurement or metric that can indicate in a human being the health status of that person, if you put that in the perspective of other people at the same age. So we know that we have a lot of these different markers based a lot on the information we have and knowledge on the hallmarks of aging and the pillars of aging. So we have a lot of different metrics that we can measure in terms of molecular and cellular mechanisms of aging, where we have epigenetics, telomere attrition, we heard about that yesterday, and also genome instability, stem cell exhaustion, and so on. And yet there are other functions, for example, loss of proteostasis, where we do not perhaps have the best markers in humans yet. And then there are also other metrics where we talk about composed scores, or these clocks, and the most famous one is, of course, the epigenetic clock that I'm sure most of you heard of, which is perhaps considered today to be the best marker or metrics of biological age in humans. But there are also other clinical metrics. We talk about disability, frailty, sarcopenia, physical function, even cognitive function, and even demographics of aging. So when we talk about biological age, it should be a marker of something that is, of course, correlated with chronological age, but it should also add something on top of chronological age. So if individuals with the same chronological age have different levels of biological age, it can tell us something about their health status. So we know that people with a higher level of a metric of biological age have an increased risk for health-related outcomes, or unfavorable health-related outcomes. And we also know that there is a time-varying trend here, so that this type of predictions work differently across the lifespan, depending on the age of the individual. Meaning that in the late life part of a lifespan, the predictions doesn't really work that well, because you can look perfectly healthy one day and then you can die the next day. So I'll just try to summarize today what we know about these biological age metrics. So first of all, we have representation of these different types of metrics in different systems, so both from molecular and cellular systems into all organisms. And they all measure a little bit different parts of the underlying biological aging process. And there is not really a gold standard yet. We don't know what's the best metric, and we don't perhaps use the same markers or measurements across different groups. Although, as I said, we still believe that perhaps the epigenetic clock is the best marker we have today. But it also relates to not only the measurements, but in the way that we model these biological age. Because it matters. We don't really want to capture chronological age. We want to capture health-related changes in life. So that's also a little bit difficult in the way that we train these models. So we need to improve the training of the models. We also know that they're correlated. So each row here or column represents one of the different metrics as an example here. So we know that they're very well correlated with each other. But if you then regress out age, meaning that if we control for chronological age of these markers, they are not very well correlated at all. So they really represent different parts of the aging process. But there are still some clusters here. So this is, for example, a functional cluster. So different markers in the functional category, they tend to capture more or less the same thing. And even so in the epigenetic cluster here, different epigenetic clocks capture more, similar parts. And as I said, we know as well that they work for predict health outcomes. So they work to predict survival, but also a lot of other diseases and health outcomes. So what we don't really know is the causation here. These are markers of some underlying biological process, but how much are they really involved in causing changes across the lifespan, and really involved in causing biological ages processes? We don't know, and we need to study this much more. So if we think about biological age and what type of interventions we can do in a normal randomized clinical trial, for example, if we want to test statins on cardiovascular disease outcome, we have two groups of treated and untreated, and because of the randomization of it into two groups, we know that, or we assume that the confounders are equal. So that means that different things that should not interfere in this study are supposed to be equal between the groups. So we don't really need to adjust for that. And then we usually check some sort of biomarker on the way. For example, LDL cholesterol level. We see that we have an as a subclinical proxy of the treatment effect. We can check on the way we check the LDL levels, how that is affected by statins. And in the end, we have an outcome, such a hard endpoint outcome, such as cardiovascular disease. So if we think about a similar RCT for aging, well, we would like to treat individuals randomized into two groups where confounders are equal. Then we would perhaps use a biological age indicator as a subclinical marker of the treatment effect, so we can measure this on the way. Because the hard outcome in aging studies is really mortality, and this is a challenge. So the idea here is that we could then instead use biological age indicators in these type of RCTs to come to some sort of conclusion earlier. Perhaps many of you saw this last week. There was a new paper out in Aging Cell, and it gained a lot of attention in media, so Nature and other newspapers picked this up. First hint that body's biological age can be reversed. So what this is really a study, a small trial, pilot trial by Steve Horvath, who invented this epigenetic clock, and colleagues. And what they showed is that if you treat, I think they had 9 individuals only, if you treat them with a cocktail of different drugs, and then you measure their epigenetic age, so the epigenetic clock indicator for biological age, and you measure it at baseline, and then 6 months, 12 months, and 18 months after the treatment. You can see all these graphs are different versions of epigenetic clocks. Basically, all of them show that there is an even decreased indication of biological age as assessed by these clocks. So this was really surprising that they did not expect this huge of a change. And this is why we see this great news outlets. I mean, it's a very interesting study because they also see something that we talked about yesterday, that even after the treatment, 6 months after the treatment ended, the biological age is intact at a lower level. So it's some sort of stability as well that they detect. So, of course, it comes with some caveats. This is a small trial. It's not a randomized trial. There are no controls here. And And the epigenetic measurements were done after the trial was completed, so it's in some way a retrospective study as well. So of course there are some caveats to this. But I think it's an interesting start or illustration of how you can think about this. So I would like to propose that I think that we can do more to bridge this gap between the animal systems or model systems of aging and human beings, because we have a lot of epidemiological data. So now we have biobanks, large biobanks such as the UK Biobank, we have China Kadoorie Biobank, Million Veteran Program in the US, and many, many more coming. And even here in Sweden, we have efforts to harmonize longitudinal aging cohorts across different sites, so we can use them in a unified way. And we also have many databases where they have information on prescribed drugs in individuals. And I think that we can use these resources in a smart way so that we can take this information and learn from what we already know is working in terms of drug use in humans. So with all this information we can learn a lot about drug repurposing. What if we have a drug that is indicating something, we just haven't pulled out the data in the right way to look at it already? So I think maybe this is something where we can bridge between the clinical setting and animal models. But let me explain some of the problems or some of the things you need to think about when you use epidemiological data. So this is ideally what we would like to know. We have healthy individuals, and we want to know the effect of a treatment. And we may have a database that we have information on individuals, and we have a lot of information on the drug use over time. Well, ideally, this is what we want to know, but very often this is what we have. So we have a lot of old people, they are multimorbid, they suffer from polypharmacy, they have lots of drugs, and it's really difficult to disentangle what is the cause and effect here, because it's so much intertwined with so many different things. And another usual caveat in using epidemiological data is what you call survival bias. And it's in pharmacoepidemiology, it's sometimes also referred to as immortal time bias. Because if you have a lot of individuals and you follow them over time, some of them will live long enough to have an exposure, meaning that they will have an effect of a treatment, just because they lived so long time, so that they sooner or later will probably start to take some drugs. Whereas others, they do not live long enough to get the exposure or to get the treatment. So this will somehow mask this association that the treatment will be beneficial just because this person lives longer, which may not be true. So there are a lot of problems with using these data, but if you are aware of them, you can learn how to deal with this and what type of study designs you need to control for it. So for example, here we have when we talk about the gold standard studies, it's really the randomized clinical trials, where we have an intervention and we have a strong causal effect from these type of studies. And in the bottom here, we have a lot of observational studies where you have perhaps cross-sectional data, and you compare diabetic patients who get metformin, and then you compare them to people who do not get metformin. Of course, that's a lot of problems in that, because you get metformin for a reason, and the people who do not get it, that's also a reason. So you need to think about smart designs in this way. But there are very many different studies designs where you can control for many things. So for example, in longitudinal analysis, you can look at individuals before and after, as we had in the example with the epigenetic clock. And you can also use, for example, twins. So if you have two twins of a pair, then you can control for an effect that you see in one twin using data from the other twin where you don't have the same effect. So in that way, you can control for a lot of factors that you perhaps cannot even measure in your data. And there are also a lot of other examples here that I don't have time to go into. But just to give you a little bit of flavor, I'll show some data on what I'm to describe this better. So for example, if you have a longitudinal study, And you do have information on all these, you have blood sampling over time, you also have information on drugs over time. You should be able to use this in a smart way. So having this information, you can of course infer a lot of different measurements of biological age. And you also have then the exposures of different drugs over time. So then ideally you would be able to use some sort of aging trajectory. So you have the individuals as some sort of self-control, because you know how they were before treatment and you know how it looks after treatment in terms of their biological age. So I'll show you one example here where we had preliminary data in one of the studies that I work with. It's a longitudinal study. It's called the Swedish Adoption Twin Study of Aging. So here we calculated biological age by a composite score of different biomarkers. So in this case, I think we used 13 different biomarkers, and we were interested to see how statin treatment affects this biological age indicator. And in this physiological age, we don't have LDL measurements. I think we have one triglyceride lipid, but the rest is anthropometrics or other types of values. So if we just look at aging trajectory in all these individuals over time, it looks something like this. Then what we can do is that we can center, instead of center the individuals on their age, we can center on their start date of the statin treatment. And then if we do that, this is what we see. So that it looks like really the before and after effect on your biological age is really changed before and after this statin treatment. So this may be, you know, some indication that there is something going on in the body using statin treatment. But of course, we haven't adjusted for many other things that can be actually important here. So another example then, if you have the same type of data, and in this study we also have twins, so we would then be able to, even within these aging trajectories, we could also compare within a twin pair. So if we have one twin that is using statins and one twin that is not using statins, we can then see, okay, so what is the difference in their biological age? Because if one twin has this effect and the other do not, we can compare. So in twins, we can compare things like the genetics. They share genetics. Depending on if they're identical or fraternal twins, they share 100% or approximately 50% of their genes. So this is a way we can control for the genetics, the shared genetics. But they also share a lot of environmental effects. So early life environment, for example, but even later life, they tend to live in similar manner across the life. So this is really a way to control for things that we don't have data on. We don't know what they ate for breakfast during their first 10 years of life. But in this way, we kind of control for it. So if we look at the same thing then, we have twins that are discordant for statin treatment. So these are the twins that take statin. So again, it's the same curve as before. It just looks a bit different. And centered on their start date of statin treatment, we can see that there is a dramatic drop in their biological age metric that we use here. So then if we look at their co-twin, of course we don't have a start date here, but if we use the same date, we center on the same date as their twin, the other twin in the pair got the statin treatment, we would then hopefully then see that there is no effect on the statin treatment here. But what we see is something like this. So there seems to be, even though it's not as dramatic change here, but there seems to be also a lot of things going on in their co-twin who do not take statins. So what is the reason for this? Then you have to start to think, okay, well, most probably the other twin in the pair perhaps took some other treatment. What we see, if the twins are discordant, we often see that if one take drugs to treat cardiovascular disease, the other one perhaps take drugs to treat diabetes. So there may be other drugs underlying this effect. There may be other lifestyle changes that is influencing. So what I mean here is that the things that we see here is probably also present here. It's just that we can't see it. So maybe the individual here, the twins in this graph, also takes other drugs, other than statin, for example. So this effect may not be completely based on the statin treatment. There may be other masked effect here that we need to control for and think about what they can be. So one way would, of course, be to take the difference between the two twins. But that may be a little bit a strong effect as well, because I think we need to more look into what type of treatments they have and how can we adjust for that. Okay, so I need to wrap up, I think. So some of my solutions to dealing with epidemiological data is then to think about, okay, what are the things that you can measure in your data? How can you control for that? But what are the things that you cannot measure? And how can you still control for that using smart study designs? For example, in RCTs, you have the randomizations. And you can do that in a lot of different ways. It's just that it's not always easy, but if you think about it, and I think also you need to do this crosstalk with translational thinking, but also including a lot of biostatisticians in the way that you model this. So just as a final example of another study where you can actually randomize people into groups is something called Mendelian randomizations. So again, this is the normal RCT situation where you have randomization into groups. In the Mendelian randomization example, you use the genotypes of individuals as a randomization, because at conception, the genes that we have, or the genotypes, are randomized, and you can use that in a smart way to measure then different outcomes depending on what genes you were given at birth. Just as an example of this is we did one of these Mendelian randomization studies where we used this randomization effect, because then we have stronger evidence of causal associations. And then we could really see that lowering LDL cholesterol or even using SNPs that mimic the statin treatment effect, we could see that we have a causal association on the frailty index later in life. So this is data from UK Biobank. Okay, so in summary, I think that we need to develop or improve the methods we have currently for biological age indications in humans. But doing that and think about smart study designs, it is possible to use epidemiological data to bridge the gap and to do studies on drug repurposing in humans. And I also think that it's really important to do the crosstalk between basic science, epidemiology, and clinics and biostatisticians in order to improve the methods and to go further in this area. Thank you very much.